Today's presentation is part of an active outreach to researchers and clinicians across the US to educate and create awareness about the potential for bioprinting technology to transform and advance your research and to be applied as a platform for evolving novel regenerative therapies. So ROKIT Healthcare is a South Korean pioneer and leader in regenerative medicine. And they are the manufacturer of some 4D bioprinters, Oregon Regeneration platform technologies, notably the INVIVO 4D and the 4D6. They have some revolutionary applications, basic and translational research and development, and autologous therapies. So, excuse me, they offer not just the 3D printers, but also genomics and single cell RNA services, among other things. So today's presentation is a combined effort between ROKIT Healthcare and Indicate Technologies. That's my team out of Santa Clara, and we also have an office in Vancouver, Washington, and ONS Bio. So that's Senthil, who will be giving the presentation today. So I will hand it over to Dr. Senthil.
Right. Thank you, Chris. Before I begin, I'll also turn off my camera just for bandwidth reasons. A very good morning to everyone in the audience. I think it's morning for you, so let me turn mine off. My name is Senthil Sarvanamuthu, as Chris just mentioned. I'm the founder and CEO for ONS Bio LLC, which is a distribution partner and consultant for ROKIT Healthcare. I thank Dr. Philip Copenhaver for this opportunity to connect with you all today and talk about bioprinting as a powerful research tool, and also about its potential as a therapeutic platform in the context of some of the emerging megatrends in science and healthcare today. In this presentation, in the interest of time, I'll be as quick as possible, and you will always have the opportunity to review these slides because we'll send out a recording. And most of the slides will also have references to some of the publications, so you can always go back and check those. Beyond the basics of bioprinting, I will also try to cover some exemplary real-life applications and case studies of the ROKIT's 4D bioprinting technologies from research labs, commercial industries around the world, and even the operating room. So as Chris mentioned, this is a joint effort by all three of our companies. And to break the ice, I want to first show a very brief video clip of the newest bioprinter from ROKIT, the Organ Regenerator 4D6, for the benefit of anyone in the audience who hasn't seen a bioprinter before. All right, so let me quickly go back. Okay, all right. So to set the context, for those of you who haven't heard about bioprinting yet, bioprinting is a perfect combination of computer science, biology, instrumentation, engineering, and material science. It brings the benefits of personalization, autologous solutions, and the fine details to human health applications. 3D bioprinting is an additive manufacturing process in which the cells, biopolymers, and growth factors are combined to create tissue-like structures that imitate native human tissues. Because these printed living tissues are often further incubated to have them proliferate, mature, and change over time, there is a dimension of time here that makes it appropriate to call 4D printing or 4D bioprinting instead of 3D So, and that's kind of what you will hear me go back and forth, 4D and 3D. But you know, when I use 4D, what I'm talking about. And we believe that it's more appropriate to call it as 4D bioprinting, especially when it involves living entities such as cells and tissues that continue to change over time during the printing process as well as post-printing. So this clip here shows the printing happening in a gel matrix, but there are more complicated, complex patterns and structures that can be printed using various techniques. But all of that falls under this big, broad umbrella of bioprinting. All right, to set the context for why you should all care about bioprinting, I want us to look around and see what's happening in the news over the last few years. There are 5 notable events and megatrends that I'd like to recall here today for the purposes of this talk that warrant our closer attention to growing relevance and the potential of bioprinting as a revolutionary technology for adoption into your biomedical research and possibly also develop methods for treatment of diseases. In September 2018, we saw the featured news of a boy who was living for 14 years with a new urinary bladder made from his own cells, seeded with 3D inkjet machines into decellularized pig bladder as a scaffold. Now, this work and similar work by many others, it highlights the promise and potential for bioprinting of human tissues to transform medicine with implications for organ transplants, cancer treatment, and many other possibilities such as better ways to develop antibiotics and drugs. So this is one of the important trends that we are seeing. Now, in July 2019, American Medical Association approved reimbursement codes for 3D printed surgical models and tools. While these are temporary codes, these are making the foundation for a clinical registry of patient 3D scan data and relevant 3D modeling, which will serve as a learning resource and tutorial, and also provide the much needed impetus for active implementation of 3D printing in biomedicine. In September 2019, United States Environmental Protection Agency announced that it will eliminate all mammal testing by 2035, becoming the first federal agency to make a strong statement and commitment to the transition to humane research while emphasizing the urgency to find alternative solutions to use of animals in research. In October of that year, ROKIT Healthcare and a diabetic foot ulcer specialty clinic in India, they put the bioprinter in the operating room to create a streamlined process of skin regeneration for diabetic foot ulcers. So this new and alternative method for chronic wound healing promotes skin reconstruction using bioinks made from patients' own adipose tissue, printed into size-specific patches and applied directly onto the wounds. These dermal patches, they integrate with the skin, and through early onset healing, ROKIT's mission is to decrease the rate of amputation for patients with diabetic foot ulcer. Now, this is critical, especially given the fact that the 5-year survival rate of patients who have undergone amputations is far lower than the 5-year survival rate of many cancer patients. And last but not the least, in the most recent months of 2020 and into 2021, we are seeing that COVID-19 has changed everything about the way we live, and most notably, we have seen the pandemic disrupt the traditional and centralized value chains in manufacturing. When hospitals got cut off from supplies of masks and ventilators because factories shut down, 3D printing came to the rescue to manufacture some of these supplies on demand and on site. Okay, now why am I saying all this? What happens if we put all these things together? What is the take-home message from these global events and megatrends in biomedicine? It points to the fact that the 3D printing and by virtue of that, many of the additive manufacturing methods, including bioprinting, can react directly to decentralized demands from life sciences research and can also serve as an effective medical strategy. Clearly, there is a growing value for on-demand biomanufacturing, and bioprinting is quite relevant in this context to meet the real world needs and these megatrends. All right, it is for this irrefutable connection to the real world that I am here today to talk about the 3D bioprinting technology. For reasons that I just discussed, it may be worth paying attention to how this technology is maturing to impact our research and healthcare workflows today. Though bioprinting is getting a lot of attention in the mainstream research only recently, the origins of the technology go back nearly 2 decades. It is not totally in its infancy anymore. In fact, the number of research publications around this technology have been growing exponentially since 2000. And that you can see here in this chart. And today there are thousands of publications as a testament to the benefits and applications of the technology across tissue engineering, biomaterial development, regenerative medicine, and so on, many different fields. So it's very important for me to point out right away that bioprinting is a platform biomanufacturing technology with applications for all major funded research fields. So it is not relevant for any one niche area of research or application. So here what you see is a list of publications that are hyperlinked. And there has already been a lot of evidence about the ideal choice and combination of cells and biomaterials for tissue-specific applications using bioprinting. And one other possibility is that perhaps in the near future, most biomedical researchers are going to come to a realization that the journals in which they publish will start requiring data from bioprinted 3D human tissues as a go between the 2D in vitro cell culture or the in vivo animal or human studies that they are so used to. And it may actually become a very important criteria for your research to even move forward. One of the major challenges in medicine, of course, is not having sufficient number of tissues and organs to replace failing or aging organs in patients. And modern tissue engineering aims to indirectly replace damaged tissue with man-made scaffolds that support cell growth. And bioprinting is at the frontier of this field because it's the only platform technology that can create these tissues and organs with applications in all these different types of diseases that come with aging. There has already been a lot of evidence about the ideal choice and combination of cells and biomaterials for tissue-specific applications involving bioprinting, and the field is progressing really fast. All right, so now I guess then it's time for me to quickly address the most basic questions. For those of you in the audience who may have prior experience with bioprinting, please bear with me. But I'm going to go with the assumption that the audience does include a lot of people who are totally new. So I'll try to quickly give the refresher on the basics. So what are the key components of bioprinting, whether it is 3D or 4D? The bioprinting technology consists of largely three components: the biomaterials, the bioprinter, which represents the hardware component, and then the design itself, which is the software component. Okay, first, let's talk about the biomaterials. Unlike the traditional 3D printer, which uses hard plastics and synthetic materials to create non-living structures, bioprinter uses materials that are biocompatible, biodegradable, and they are derived from both natural or synthetic sources. Common examples of naturally derived biomaterials include collagen, gelatin, elastin, chitin, coming from the extracellular matrix of human or animal cells. And they also include alginate, sericin, or cellulose coming from seaweed, insects, plants, respectively. And natural hydroxyapatite comes from mammalian bone. Synthetic biomaterials are biodegradable polymers with both strength and elasticity to mimic human tissues, and some of these have been put to good use in FDA-approved medical devices. And finally, you have also the composites, which often combine natural and synthetic materials to add or enhance certain mechanical or chemical properties. Now, these biomaterials can be soft, low viscosity hydrogels or hard biodegradable plastics. And when hydrogels are combined with cells for printing, they often get the name bioinks. Bioinks, they basically serve as the extracellular matrix when you are trying to print cells outside the body. And bioinks offer protection from shear stress during the printing process. And then they provide the cells with nutrients and hydration, and they give the much needed overall physiochemical support to maintain the structural integrity of the printed tissues. So when selecting the materials for bioinks, there are several key criteria, but they can basically be summarized into two broad categories. One, the bioinks should be able to extrude out of the nozzle tips and stack layer by layer, structurally speaking. Now, and this can happen, and this needs to happen with or without post-printing processing. It depends on the nature of the material. The second thing is the bioinks should be biocompatible and they should uphold high cell viability. Now, these are the important criteria. And as you can see here, there are a bunch of different properties that need to be taken into consideration when choosing the right material for your bioprinting application. Okay, now let's talk a little bit about the hardware. There are various methods of bioprinting today, as shown here. They range from inkjet-based approaches to laser-assisted and stereolithography methods. But among these, extrusion, which is highlighted here, is the commonest. Nearly 70% of the bioprinting, you could say, is done using extrusion-based methods. And our bioprinter from ROKIT is also an extrusion-based bioprinter. And one key component of any bioprinter hardware is the print head that should be able to print both soft as well as hard materials. There are two ways that the print heads can actually go about doing this. One is in a scaffold laden way where you print hard matrix, and the other one is scaffold free, which does not have any hard matrix. And another key component is the chamber's ability, the printing chamber's ability to mimic a biosafety cabinet, because after all, when you're dealing with living cells, you need to account for optimal conditions within the printing chamber. So to optimize cell viability and functionality under the stress of printing, the chamber should have UV disinfection, HEPA filter, and some type of bioreactor capability to provide the optimal environments for the cells, especially when it comes to temperature and humidity control. And finally, we've got the third component of bioprinting platform, which is the design component, meaning the 3D models of the objects or the tissue types or the organs that we want to print are the digital files, the 3D model files. They are often known as the STL files, can be obtained either by manual creation using a design software, or they can be derived from a CAD, MRI scans of physical structures like human bodies or organs. Now, these STL files then get processed with a slicing software into a standard machine language file that describes how a 3D printer should print the job in a given three-dimensional space. It includes the information on the XYZ positioning coordinates and the printing speeds and what type of infill patterns, etc. And there is a whole bunch of parameters that you can talk about relevant to the bioprinting. Now, these examples here that you see in the bottom help you visualize some of these. For example, you will see that the STL object and G-code variations can be seen here. This is the whole object and how the fine infill patterns can vary between, depending on your needs. All right, so now that we understand the components of bioprinting, we can put all this together logically and then try and see how the process flows. So the process involves 5 key steps. To summarize briefly, first we got to do the preparation, which involves designing the experiment with a clear identification of the problem to address. And it includes the selection of the right biomaterials, cell types, and 3D models that the printer will use. And then you got to do the printing, that is the actual printing process. The 3D models are then sliced into appropriate printing instructions through the software. And the bio ink is placed in a printer cartridge, and the deposition takes place based on the digital model. And then we have the post-printing, where you take the printed structure and do a mechanical or a chemical stimulation so as to create stable and functional structures for the biological material. And the post-printing process may include cross-linking by either thermal means or chemical or light induction, and it may involve a differentiation of the cell type, a whole bunch of things that you can think of. Evaluation is a very important step because this is the step where the culture printed tissues are assessed for structural, morphological, and functional characterization in comparison to a gold standard living tissue, and see how well we are able to biomimic the actual tissue that we set out to print. And application involves the use of the bioprinted tissue for various purposes, and that could range from disease modeling to in vitro safety and efficacy testing, or the holy grail would be to be able to print and use the printed tissue for transplantation into to replace failing tissues or organs. Now, to understand this process further, here I have taken the example of cardiac patch engineering and see how these different steps that we went over would actually translate for this particular case. And in the interest of time, I'll just skip this, but it's very intuitive, and you can always come back and revisit this slide when you have the recording. But I'm sure you get the idea of the workflow. Okay, so the cardiac patch engineering that I just mentioned is just one of the many downstream applications possible with bioprinting. And as I mentioned before, bioprinting is a platform technology with many, many different applications. Bioprinting has been used across tissue engineering, biomaterial development, regenerative medicine, veterinary medicine, toxicology, pharmaceutical sciences, neuroscience as well, and so on. So it is very important for you to realize that and understand that this is a powerful tool that you can adopt to your type of research, and it's not necessarily meant for any one or two specific applications. For simplicity, I can summarize all of these different applications into three key areas. One would be the in vitro efficacy test models for toxicological testing and screening of novel products, or whether it be a drug, cosmetic, or food. And this is going to be especially important in the context of replacing any animal testing. And the number two would be the in vitro study models for human tissue physiology and disease modeling. And lastly, in vivo models for transplantation and regeneration. And let's take a little more of an in-depth look at these three different categories, broad categories. So the in vitro efficacy test models for toxicological testing and screening of novel products. An ideal study to evaluate any toxicity of a chemical or a substance to humans would require an extremely large number of human subjects to represent the diversity of humans. But this is unrealistic and outright unethical, we know. So the use of human relevant tissue models that are derived by bioprinting can offer an alternative and a good bridge between the human clinical trials and the animal studies with a standardized human tissue fabrication process and a high throughput specific deposition of the test chemicals. And here you can see the example of bioprinted human tissues that are used for toxicological testing, novel drug development, and how the printed tissues stratify and look similar to the in vivo derived tissues, and how they also express the key differentiation markers. All right, so the second broad category of applications I mentioned is the in vitro study models for human tissue physiology and disease modeling. Traditionally, science has relied on 2D cell culture and 3D spheroids, but decades of research have clearly shown that human tissues are hypercomplex and heterogeneous and structurally, mechanically, and biochemically speaking. So it is not possible to equate a 2D cell culture or a 3D spheroid to a in vivo tissue. So cancer and tumor is a great example of that complexity, and bioprinting's ability to fabricate and visually map tissues in 3D that enables us to better and near accurately understand human body as well as disease. And as a consequence, it can also help us develop accurate therapies for complex conditions. The last category of applications is the in vivo model for transplantation and regeneration. So one of the most common challenges in medicine, of course, is not having the sufficient number of compatible tissues and organs to replace in the events of a trauma or organ failure. So flat structures like skin and tubular structures like blood vessels and urethras may be relatively simple to get started on bioprinting. But the hollow non-tubular organs like bladders or pumping organs like the heart and lungs are very difficult to replicate. They are really highly complex. So for such high complexity organs to be created, bioprinting technology can provide a certain degree of precision that surpasses any other technique or human hands. And so if these could be made with patients' own cells, autologous therapies, then the challenge of immune rejection and donor organ shortage problems that we see when we speak of organ transplantation, this can also be resolved once and for all. So here, I'd like to show you a video of a beating heart. Now this is a bioprinted cardiac patch that beats like a real cardiac tissue. So as an extension of this same dream of being able to print tissues for transplantation, it's possible for us to even predict that in future there might be advances in nanotechnology and smart material incorporation that can be combined with bioprinting that can lead to more smarter organs that would have extended durability that would be unimaginable for any naturally occurring tissue or organ. All right, so in the context of all of these things that I have discussed so far about bioprinting, ROKIT Healthcare is emerging as a key player, an industry leader in bioprinting and the organ regeneration space through innovative products, services, and robust in-house R&D programs that are aimed at creating breakthroughs in science and healthcare. Now, ROKIT is also dedicated to providing these innovative offerings to well over 25 countries around the world with over 250 user institutions and partners. Now, as a leader in this field, ROKIT introduced the state-of-the-art bioprinter, Dr. INVIVO 4D, which you see here, back in 2016 as the world's first clean chamber bioprinter with built-in HEPA UV lamps and hybrid print heads that can handle both soft tissue printing as well as hard tissue fabrication. And more recently, they also have launched the upgraded advanced medical grade material incorporated Dr. INVIVO 46, which is the world's first bioprinter and organ regeneration platform to fully integrate a cell incubator and a plasma sterilizer, along with 6 print heads for biofabrication. So one can take it to the tissue, the whole process to a next level. And I'll show more about these features and capabilities, if time permits, towards the end of the talk. Now, let's get to the most important part of today's presentation, which is to look at applications and let's try and get started with the applications that I understand would be of most interest to you all, which would be the neuronal tissues. But prior to that, let's see, basically this slide summarizes the outreach and the different areas of activities at ROKIT. So talking about the neuronal tissues, which I understand is of most interest to the bond group here. The peripheral nervous system and central nervous system, about which I'm sure you all know way more than I do, are some of the hardest tissues to regenerate and repair. Now, in case of the peripheral nervous system and the central nervous system injuries, traditional neural tissue replacement strategies, including autogenous nerve grafts or nerve graft substitutes made with autogenous vessel, muscle, and decellularized autografts. Now, the mismatch between the size and length of the donor and recipient nerves can be a problem. It can lead to very poor regeneration capacity. And we also know that these are some of the challenges that we find today. Now, for all these reasons, the use of artificial nerve guidance conduit made with biocompatible materials is kind of on the rise. But with the traditional tissue engineering techniques, such as, let's say, electrospinning or scaffolding, there is very little control over the complexity of the architecture. There is difficulty in that space. So we have to look for other effective methods to fabricate these tissues. And that has kind of been an unmet need for a long time. So to overcome these limitations, 3D bioprinting has been rising to the table to offer an ability to confer ability on the users to make free-form designs with various types of cells and biomaterials. Now, despite the fact that its application in PNS and CNS regeneration is still very new, it has made progress over the years. And this figure here kind of summarizes where these applications currently stand along the developmental stages towards clinical or commercial use. So on the left, you see that the scaffolds for central nervous system repair are still at a very early stage in development, as are the 3D printed in vitro brain models. Now, towards the far right, you see the 3D printed scaffolds for peripheral nerve repair that are slightly further in development, as are the in vitro models for peripheral nerves with relative simplicity. Now, in the next few slides, I'll show you some key examples from journals over the last 3 years that have demonstrated advantages of bioprinting in modeling nerve tissues. In modeling the peripheral nervous system, a main challenge is the incorporation of multiple cell types into specific patterns. Now, in this context, 3D bioprinting offers the advantage of positioning cells in multiple compartments or microgrooves and thereby aligning them the way they should be for optimal axon growth. For instance, as shown in this figure, this is work done by Johnson et al. Research group. What they have done is they have created an in vitro device for studying the peripheral nervous system that resembles a mini nervous system on a chip. So this 3D printed device contained 3 chambers, one for the peripheral nervous system cells, and one for the Schwann cells, and one for the terminal cells. Now, in addition, the device contains 1 parallel microchannel made from silicone and 2 a sealant layer right over them that you see here. Now, the reason they do that is the Schwann cells and the terminal cells, they interact with the neurons and each other solely via the axonal network guided by the silicone microchannels. Now, if you take a look at how the design on the left has translated to the 3D printed result on the right. Now, in this article, it's very hard for me to really get to the fine details of this, but as a take home, all I'll say is the authors not only demonstrate the basic cell viability and structural mimicry of the tissue, they also demonstrate the ability to track viral infection as it spreads from one cell type to another within multiple chambers of the model PNS device that they fabricate. This is just one example, and the publication is here. You can always refer to that later on for more insights. Now, on the other hand, modeling the central nervous system has far-reaching implications for studying brain and spinal cord function that would otherwise be difficult to examine in vivo. Already over a decade ago, several research studies have shown that printing human neural stem cells into 3D structures and then differentiating them into mature neural cells led to very high expression of differentiated neural markers than the same cells that are cultured in two-dimensional cell culture, 2D cell cultures. So the same was also seen in the iPSCs following the same process. Bioprinting in 3D also permits the creation of very complex structures that recapitulate important aspects of the brain structure. For example, the human cortex contains a distinct 6-layered structure. We know that. Now, recreating this structure that you see here in a controlled manner is difficult to do manually. It's simply impossible. But it can be achieved efficiently through 3D bioprinting because of the ability of 3D bioprinting to offer that level of precision in control of what cells and the layering and many other parameters. So in this study that is kind of summarized in the figures, the group used a bio ink consisting of a novel peptide modified biopolymer, which is a gel-an gum based bio ink. This was combined with primary cortical neurons, and then the gel-an gum, which itself is a polysaccharide obtained from microorganism fermentation. It was first introduced as an alternative to agar and gelatin in the food industry. Now, the tissue engineering gel-an gum has shown many benefits over the other hydrogels, such as low production cost, biocompatibility, and achieving similar levels of mechanical strength as a gelatin or agar at lower concentrations. So the take home here is that in this study, the research group well demonstrates that they can create 6-layer brain cortex structure, whether in vertical or a planar orientation. Although still at an early stage of development, this example clearly highlights the potential of 3D bioprinting for creating complex 3D in vitro models of the brain. Now, by no means these are advanced enough, but these are early promising experiments that one can take and combine it with the platform technology, the power and precision of bioprinting, your own niche knowledge of your specific areas and the biology of the cells that you're dealing with, and combined with an ever-growing list of biomaterials and all that. Now, when all these three things come together, then I think there is enormous possibilities for many new innovations, both for creation of new research models as well as potential therapies. All right, so this publication, I'll go quickly, is a publication by one of our own Dr. INVIVO bioprinter user in South Korea at a government level, Korean Institute of Science and Technology. So in contrast to the in vitro models that we discussed before, this one showcases the use of bioprinting to create regenerative strategies for in vivo peripheral nerve injury sites using a nerve guidance conduit. Now, in this study, the research group developed a biocompatible elastic NGC. They first made nanoporous membrane of PLCL with electro spinning. And then they rolled it up and designed the interior with neutralized or oriented collagen hydrogel patches that were printed in high resolution using Dr. INVIVO 4D2, which is the first generation bioprinter. So by the way, PLCL is a very widely used biomaterial in applications such as wound healing, formation of cartilage, cardiovascular applications, because it has excellent biocompatibility and biodegradability, as well as a very high elasticity and flexibility compared to the regular PCL, which is polycaprolactone or PLA. Collagen also is a widely used bioink, we all know that, because it makes up a large percentage of even the natural ECM in our body. So this group compared the efficacies of the PLCL conduits filled with bulk collagen and the 3D printed collagen with the autogenous nerve graft for peripheral nerve regeneration. And what they find is that the in vivo study using the sciatic injury model in rats, the 3D printed nerve conduit guidance group exhibited denser and more organized myelinated axons than the group made with bulk collagen hydrogel. So the group concludes that the that the aligned collagen hydrogels they enabled the alignment of the collagen hydrogels is enabled by the 3D printing patterns of the groups, and it appears to provide a preferential environment for the nerve regeneration, and it provides a guidance path along the longitudinal direction. Now, this was also true for both the axonal regeneration, remyelination, as well as the functional recovery. Now, why I'm saying this is a great example of how bioprinting can help with regenerative tissue engineering by providing an architecturally suitable environment inside the conduit for axonal growth. Now, given that given that traditionally speaking, Schwann cells have a very poor ability to migrate and align through the gaps. The ability to make the paths by designing the interior with the bioprinting will give a kind of a crucial advantage to an effective axon regeneration across this gap. More details you can always refer to the original paper, but so all I'm trying to emphasize here is that the use of bioprinting is beneficial in some of these efforts using the neuronal tissues. All right, so as we could see in summary, as we could see from these examples of in vitro neural tissue models and nerve systems, bioprinting offers three major advantages. One is the bioprinting offers rapid free-form prototyping capability, allowing for a faster fabrication of living tissue constructs of diverse 3D topologies compared to the existing tissue engineering techniques. Now, second, it also has an excellent ability to construct tissue models with a homogeneous cell distribution and a uniform spacing. Now, that gives a clear advantage of repeatability of the experiment. And lastly, bioprinting also provides exceptional versatility in cell positioning, whether it is vertical in full layers or planar in different cell compartments. You have the ability to control those. So these are all crucial factors to study a living system with complex biochemical and interactions and architectural features. And so in that context, bioprinting is quite promising and definitely worth considering for your own research. All right, I don't know, we probably don't have much time, but I'll quickly go over some of the, or at least mention some of the other applications in slides, in the next set of slides. So you see how truly it's a platform technology with wide-ranging applications. So in this example, without going into the nitty-gritties, the investigators ended up using INVIVO bioprinter from ROKIT to make a self-healing hydrogel, which has the potential in tissue engineering applications. Especially in this, this particular group thinks that the self-healing hydrogel can be applicable in the cartilage regeneration applications. And this is a case of the application of bioprinting for adipose tissue regeneration. And this is an example of of the work done by the Seoul National University Research Group. They sought out to engineer a novel way to regenerate adipose tissue, and they used conventional medical grade materials such as PCL, which we know are not they don't match the mechanical property of the adipose tissue. So they made a modified version of that called the PLCL and mixed it with adipose derived ECM. So the group was able to show that when implanted into the mice, the size customized scaffolds, they promoted adipose tissue regeneration with macrophage infiltration and vascularization. And the group confirms that bioprinting combined with the new complex scaffold that they have developed can offer a new solution for burn, trauma injury, and post-mastectomy repair, especially in large defects. Here is an example of the work done using the INVIVO bioprinter by the Fraunhofer Institute in Germany. They developed a patented disc that allows the iPSCs differentiate into cardiomyocytes to align and create a beating model of the heart. And I can show you. Now, what's particularly interesting here is the fact that the bioprinted heart patch has been beating at least for 6 months in vitro. So that talks a lot about the viability of the bioprinted tissue. And here is an example of the application of bioprinting to generation of muscle models for drug testing. The research goal was to develop a 3D in vitro model to mimic human muscle tissues. And that's what they have been able to do here and apply to drug testing. Now, this is an example of creation of disease models that can better predict drug responses. And cancer is one of the hottest topics in this area. As we all know, existing in vitro models of cancer are often single cell spheroids, and they lack the tumor microenvironments, which is a big deal in this field, right? We all know that the tumor microenvironments, TME, they are crucial to the intrinsic behavior and malignancy. So in this example, we've got bioprinted layer of lung fibroblasts and HUVEX, human vascular endothelial cells in bioinks of alginate, fibrinogen, and gelatin. And then the multicellular tumor spheroids of glioblastoma cells are seeded onto the vascularized tissue and incubated, resulting in a vascularized tumor model. Here is a case of bone application where synthetic HA, hyaluronic acid, reinforced with strontium, a metal ion that allows pure, hydroxyapatite. Hydroxyapatite to dissolve better and induce optimal formation of bone tissues in the early stage of implantation. For the first time in this study, the strontium HA bioceramic scaffold, they were printed with 3D printing technique using the Dr. INVIVO 4D bioprinter. And in vitro bioactivity tests demonstrate that the scaffold printed with the strontium of a certain strontium content, it promotes rapid cell proliferation and osteogenic differentiation and cellular mineralization. This is an interesting example of how interesting the applications of bioprinting can get. So here is an example of a QR-coded drug system, drug delivery system developed in the form of thin orodispersible films, which can support patient compliance because they are not only easy to swallow, they are also highly informative, and they deliver information about the patient's prescription right upon scanning of the drug itself. And this can be or this has the potential to be used as a way to reduce misconsumption and misadministration of medicine. And this is one of the trend these days by pharma companies that are trying to replace anti-cancer injections using orodispersible films, which might be a more effective and minimally invasive way of doing it. This is an example of application of the bioprinting to develop an egg-shaped bioresponsive device that combines microbes, hydrogels, and conductive carbon nanofibers. The reason why I have this example here is the application doesn't have to always be involving only mammalian cells. So it could also involve microbes, as you can see in this case of development of a biosensor, so to speak. So the unique advantage of 3D bioprinting in this context is that the ability to inject microbes and carbon nanofibers at the desired positions in desired locations. That proves to be very useful here, because the group was able to position target cells and gather their desired responses in a controlled environment. And the group also suggests that the microbial bioresponsive device can be a useful one for studying wastewater treatment, microbial biosynthesis, metabolism, and photosynthesis, and so on. All right, so I've talked, there are a lot more examples, and it's simply impossible to cover all of that in a 1 hour talk. But the last bit I want to emphasize is, all these interesting applications that are at the bench side, they are great. But the most noteworthy applications that we all look forward to or want to see more of are the ones that take bioprinting off the bench into the clinic. So as an effective medical strategy, how is it being used? And as I mentioned before in the beginning of the talk, ROKIT Healthcare has been putting a lot of emphasis on this, on moving the bioprinting applications to the bedside for patient benefit. And so in the last 2 years, they have done a lot of good work in the area of diabetic foot ulcer as one of the immediate applications of bioprinting. As we all know, diabetic foot ulcer is a chronic condition with side effects affecting almost 25% of all diabetic patients. At some point in their lives, they suffer diabetic foot ulcer or similar skin issues. So more than 20 years, the traditional wound care system has totally relied on dressings and collagen patches and negative pressure wound therapies, autografts. But none of these techniques have been truly autologous and regenerative. And so ROKIT took advantage of bioprinting and has developed a bioprinting-based skin regeneration process in which the bioinks are the extracellular matrix and the microfat particles obtained from patients using liposuction. And these bioinks derived from the same patient are printed into specific wound-specific patches, and those patches seem to have tremendous regeneration effect. In fact, in a clinical trial, nearly 100% of the patients ended up having a complete epithelialization of the wounds. And these are not any ordinary wounds. These are wounds that were left unhealed or couldn't be treated for months to even years. And with the application of the bioprinted liposuction derived patches, almost all the patients had 100% epithelialization within 4 weeks. So such dramatic was the effect, and they are pursuing more regulatory clearance to bring this into the clinic. This is a little bit of the workflow here, but I'll skip that and I'll show you this quick video where it's possible for us to conceive that in future, as this technology gets more mature, and perfected, it's possible to have a synchronized workflow where the wound is scanned, and then it is digitized, and then the software calculates all the parameters, and then the patient's autologous material is harvested and then bioprinted into custom-made patches, like you see here, and then they can be immediately applied onto the patient right in the operation theater to bring about a nice therapeutic solution. And ROKIT is trying to cover the entire workflow. Okay, so I've gone through extensive array of applications across many disciplines. The key point here is that bioprinting is a platform technology. It's not restricted to any one discipline or cell type or biomaterials. So basically, sky is the limit when it comes to its application potential, and you can be as creative as possible in your own research efforts to adopt this and then come up with your own version of the applications. Now, in the last few minutes, I will quickly, now this slide shows you the kind of the different types of institutions and the partners that have currently, that fall into the INVIVO bioprinter user group. And this also represents a huge body of knowledge that also you could rely on to gain key insights and also learn from and collaborate as you embark on your own applications using the INVIVO bioprinting technology. So I'll just quickly go over the key specs and show a little more of the working of the bioprinter, for also for the benefit of those of you who may not have seen a bioprinter before. This is the INVIVO 4D bioprinter system that was launched in 2016, and when it came out, it was the world's only desktop bioprinter to have the inbuilt biosafety cabinet type environment and the HEPA filter and UV sterilization. And it also happens to be, with the advent of this DFU project that I just mentioned, it also happens to be the first bioprinter to have been used in a human clinical trial study. There are over 250 references and 30 plus publications and 250 plus labs that currently have the INVIVO bioprinter across the world. And the bioprinter is able to handle all kinds of materials, ranging from bioinks, powder mixtures, hydrogels, synthetic polymers, pellets, powders, pastes, all kinds. I'll show you just clips of how user-friendly it is to load, and these use the standard BD lower lock syringes, no fancy consumable. So you can, so they are quite compatible with whatever is out there. And these are the pneumatic hot melt dispenser for the pellets and powders, such as the PCL, PLGA, that may be particularly useful for, say, bone-like application, hard tissue applications. One important point is, based on the feedback from the users and also with the inherent goal of being able to make this as a true organ regenerator, some of the unmet needs were identified. For example, the human body is a curved surface. So you can't directly apply a biomaterial to a curved surface without the ability of the printer to do in situ 4D printing on a curved surface. So the other thing is, when you are going to develop a translational therapy, it is important that all substances are treated potentially infectious, and the device should be able to sterilize itself, and should so the sterilization component is important to consider. And lastly, since we deal with mammalian cells that grow best at their native conditions, control of temperature and pH and carbon dioxide tension and humidity is very important. So taking all these into account, the INVIVO 4D bioprinter, which is more a research grade printer that, in its original conception, all these features were incorporated, and thus was born the most advanced organ regenerative INVIVO 46. So this is the newest version or iteration of the bioprinter from ROKIT. It has a rotary head with 5 different syringe dispensers and a fixed print head for the hard plastic material or pellets. And what this means is you have got the power of 6 print heads to apply and devise complex architecture, as you can see, that you may need in fabricating complex tissue like retina or bone or muscle or heart and any number of tissues. So that's a very useful feature and a very important one too. And then, as I mentioned before, since we use human cells and tissues, the incubation, sterilization functions are essential and critical, and all of those are incorporated here. Now, this is an intelligent bioprinter system that can also tackle high throughput applications. So it can print onto even 384 well plates. And this becomes especially important in the context of applying the bioprinting approaches to development of many drug development, many organ drug development workflows, as well as in vaccine research or PCR genotyping, which demand high throughput workflow. As I mentioned before, the variety of different print heads provides you huge ability to tackle huge ranges of temperature and pressure, and kind of this makes sure that this gives the hybrid capability of different print heads to tackle any type of material that you may be currently using, as well as make it somewhat future-proof as more novel materials, biomaterials are introduced. And lastly, it also is CGMP ready, and it helps you, it kind of puts you on a fast track to medical commercialization of your innovations. And it has a very intuitive interface for controlling, either from a mobile device or a tablet, and it has the ability to be controlled across many different means and platforms, as one would expect these days. Okay, so now I guess it's time for me to conclude this talk. So I want to give a little bit of a take home. And the important thing is, at the end of the day, the take home message from this session is that bioprinting brings 3 breakthrough benefits to research and novel therapy developments. Automation, faster and precise, physiological relevance of the printed tissue, and on-demand fabrication. Now, what these breakthroughs result in? Ability to experiment with better repeatability and better physiological relevance. Ability to generate biomimetic structures and tissues without having to depend on model systems that are either too expensive or simply not ideal. And the ability to create more sustainable research methods, phasing out animal models with alternative human cell-based methods of testing. Now, one can imagine that as an extension of all these fast-changing developments in the field, one can imagine establishment of bioprinting centers of excellence across the national and international levels. And in fact, it so happens there are already quite a few of those emerging. This is to show that the field is gaining a lot of traction across the world among researchers and hospital systems. And lastly, I want to leave with this message that the basic premise here is that the era we live in today is an era of regenerative medicine. And in this times, bioprinting is not only a revolution that's underway for the way we create and use model systems for research or for answering biological questions. It is also revolutionary in the way you practice medicine. Instead of using the systems and approaches that rely on treatments of the symptoms using small molecules or monotherapies, bioprinting shifts that focus towards harnessing body's own healing potential that's inherent in the stem cells. And this is also the vision and mission of players like ROKIT. So the future of medicine is already being realized today by some of these pioneering technologies across various disciplines of science. And I'm really happy and thankful that you all share the same interest and passion. I thank you all for your time today, and I will be happy to take any questions, and thanks for your patience if I went overboard with time.
Well, thanks very much for a very interesting presentation. We did lose some people along the way because of the time, but I really appreciate you taking time to go through this. Do we have any questions from our remaining participants? I suspect you'll be getting more by email later on.
Sure, anytime.
I think people are in the midst of digesting all that you sent us.
Yes.
You said you're going to send us the link for the recording. And will the slides be embedded in that link?
Yes.
Great. Glenn, did you have a question?
I have a question.
Yes, please.
So one of the biggest problems with doing any type of application where you take human stem cells, neural stem cells, and try to run them through devices is shearing.
Oh.
You mentioned something about neurons, and I have never heard of anybody dispensing neurons through a needle and actually winding up with viable cells. Did you mean neural stem cells?
Actually, it so happens that not the neural stem cells. Most of the stem cells that we have seen in publications are either the MSCs or the human embryonic stem cells. But I'm not aware at this point of time, but I'll double check if there are publications that have also used the neural stem cells directly. As I've said, you are all the experts in this, and you probably.
So I work with human neural stem cells and also with iPSC derived cells as well.
Yeah, iPSC is no problem. Because
iPSCs should be very easy to do.
Right.
But the more differentiated cell types are the more types of cells that you want to do for essentially building basically something like a brain.
Can you tell me what the size ranges of a neural stem cell would be in terms of microns?
So I believe that the ones that we work with are on the order of, say, 10 to 20 microns in size.
So if that's the case, then the shear won't be a problem, because the needles that we normally, meaning the printing needles that we use to extrude the material are 100 microns, 200 microns, somewhere in that. You can choose like 100 to 200, and a lot of people use 200 as kind of a default. So then I think the size, if they are only 10 microns, should not be a problem for them to pass through the needle. So it may come down to optimizing and standardizing the pressure or the type of bio ink that keeps them happy during the process of harvesting, during the process of printing and post-printing. So in fact, this might be a good trial experiment for someone like you to see.
Well, what's interesting is we have human neural stem cells from individuals with healthy controls, age-matched healthy controls, and neural stem cells from individuals that have a rare prototypical neurodegenerative disease with features of ALS, Parkinson's disease, and an Alzheimer-like dementia.
Oh.
We have the iPSCs as well, but we've been able to generate neurons and also astrocytes from the neural stem cells of the subjects.
Because I'm looking at a lot of papers here, actually, as I'm speaking to you, that do use the iPSCs, the HESCs, as well as MSCs.
I saw, I think I saw on one of the citations for the individual types of tissues, organs, that essentially in the neural tissues, you had NSCs, which are neural stem cells.
Neural stem cells. Okay, then that most likely is what they have used. I'll go back and check those papers, and then you can also please check, and if we need to discuss that further. Yeah. If it's one of our own user base, then we can connect you to them.